From Louisiana State University in Baton Rouge, Louisiana. My name is Rebecca de Jesus Crespo, and I'm an assistant professor in the Department of Environmental Sciences at LSU's College of Coast and the Environment. It is my pleasure to serve as the moderator for today's event. Earlier this month, we posted a webinar on YouTube titled The Relationship Between the Environment and the Coronavirus. Given the public's interest in the topic and the fluid and constantly changing situation with COVID-19, we wanted to present a follow-up. In today's webinar, we will discuss scientifically the essential components necessary for returning to the workplace. Before we begin, I should let you know that the statements, views, and opinions shared in this webinar belong to our panelists and are not those of Louisiana State University or the LSU Board of Supervisors. Our webinar today will begin with Dr. Rebecca Christofferson, who will discuss the strengths and weaknesses of mathematical COVID models. Following that, Dr. Joel Baines will talk about the limitations of and information provided by testing of SARS-CoV-2 antigens and antibodies. Then we will conclude with information on antiviral drugs and vaccines from Dr. James Diaz. All of our panelists are well-respected experts in their field. Allow me to begin by introducing Dr. Rebecca Christofferson, an assistant professor of pathobiological sciences at the LSU School of Veterinary Medicine. Dr. Christofferson received a Bachelor of Science in Zoology, a Master's in Applied Statistics, and a Doctor of Philosophy in Arbovirology, all from Louisiana State University. She was a 2018 TEDx LSU Selected Speaker and received the 2018 Suedes Research Award for Veterinary Research Excellence. Dr. Joel Baines is the Dean and Dr. Kenneth F. Burns Chair in Veterinary Medicine at the LSU School of Veterinary Medicine. Dean Baines received a Bachelor of Science in Microbiology at Kansas State University, a Veterinary Medicine doctorate degree from the University of Pennsylvania, and a Doctor of Philosophy from Cornell University, where he studied feline coronavirus while working on his dissertation. Dr. James Diaz is the Professor of Public Health and Preventive Medicine and Director of Environmental and Occupational Health Sciences at LSU's Health Sciences Center in New Orleans. He earned several degrees from Tulane University, including a Bachelor of Science, Doctor of Medicine, Master of Health Administration, Master of Public Health and Tropical Medicine, and Doctor of Public Health. So without further ado, I present our first expert, Dr. Rebecca Christofferson.
Oh dear. So I'm going to give you an introduction to modeling for pandemic response as it relates to SARS-CoV-2. So mathematical models have been used for nearly a century to explore the dynamics of infectious diseases. And one of the most widely used metrics stemming from these models is the basic reproductive number, which you will often see depicted as R0 or R0. This is a measure of the spread of a pathogen in a completely susceptible population. It's a number of secondary cases arising from a single infectious person. So if I take a single infectious person and I introduce that person into a crowd, how many people in that crowd will become sick? And that's R0. In the SARS-CoV-2 outbreak, R0 has been estimated to be anywhere between 1.5 and over 5. This variability is due to the specific context in which it was calculated and the underlying assumptions of models from which R0 was derived, and I'll speak more about assumptions in a minute. It's also important to realize that the definition of R0 does include the spread potential of a pathogen in an entirely susceptible population. So this means that once immunity begins to build, R naught is not the most appropriate metric to use. So then we start talking about the effective reproductive number, which is sometimes seen as R sub T or R effective or just R. And this should be used because it accounts for building immunity in the population. The caveat here, of course, is that you need good estimates of immunity, and so you need good tests to assess estimates of immunity in the population. So in my opinion, there are two purposes of models being used in outbreak science. The first purpose is to forecast, of course. These are often statistical models that are driven by probabilities that are informed by the most current data available. So we've seen a lot of models out there that are constantly being updated as more data becomes available. The second purpose is to quantify outcomes of simulated scenarios, such as interventions or timing of testing, etc. These purposes are not mutually exclusive. Some models use scenario modeling to forecast the what if, and we've been seeing that quite a bit lately. And then what if we relax our social distancing? What if we go back to work too early? Each of these types of models has its own set of values and limitations. So I'm going to start with the limitations and caveats of models. So it's important to understand that the overall purpose of mathematical models in general is unique to each model. So every model was inspired by a hypothesis, much like an experimental design or a clinical trial is designed to ask a particular question about an experimental system or a therapeutic. Overall, models need experiences and human insight to guide them, just like medical treatment should not be informed solely on molecular tests. In the absence of clinicians' interpretation of them, models need context. Another misconception is that model complexity and utility are a one-to-one ratio. More complex models usually mean that there are more parameters, which are the moving parts of a model and the machinery of a model. These parameters each have assumptions about them that, one, they are informed by good data, two, they are appropriate for the model, and three, that they follow a certain distribution. So parameters themselves are rooted in statistical probability, which is rooted in data, which is why data flow is so important in data and good data. So there's a popular saying in modeling, garbage in, garbage out. If I parameterize a COVID-19 model, for example, with all of the available information I have about rates of transmission of a slow-growing bacterial pathogen, then my model will be garbage, because that is obviously not an appropriate way to parameterize this model. But if I parameterize a model of a novel respiratory pathogen with all of my insights about similarly transmitted pathogens, as was done early in the epidemic, my model will have more credibility and more utility. And then as the pandemic progresses, more and more so SARS-CoV-2 specific data is available to appropriately inform models. And so our models become more and more precise to the actual system that we're trying to project. So that being said, things are changing every day. In Louisiana, and for example, our models were initially informed by the same data coming from Wuhan, China, and Italy especially. We were doing the same types of investigations into healthcare system stress, projected morbidity and mortality, trying to predict or forecast stockpiling and resource allocation. But then we started to see the data regarding comorbidities, and some of us realized quite quickly that our at-risk population in Louisiana was going to be a bit larger because we had quite a high rate of those comorbidities. So this meant that our healthcare system was likely to be more stressed because of population potentially needing ICU beds or critical care. And so at that point, the assumptions of our models had to change a little bit to be context specific for the population of Louisiana. So models must be updated, again, on a context specific basis in order to apply to specific settings. There is no one model fits all. Another limitation or consideration, rather, is the scale of the model. A model for a hospital isn't going to be the same structure or parameterization as a model that you want to apply to a city or to a country or to a nation. So scale is an important consideration when you think about how models are interpreted and how they're parameterized and built. One of the biggest, one of the weakest links, I think, in modeling is the communication of the assumptions that underlie a model. For example, if we were to assume a model if we were to assume severe disease risk is the same across all demographics, that model would not be appropriate for COVID-19. Likewise, if you assume that the rate of transmission is limited only to known cases, then that's also inappropriate in this scenario. Some models, assumptions are less obvious. For example, a common model assumption is homogeneous mixing. And what this means is that every individual in a model has the same probability of interacting with every other individual in a model. This assumption does not mean a model is wrong. And many informative models make assumptions like this. But if your goal, for example, is to track cases in the context of social distancing adherence or lack of adherence, then when you look at social distancing behaviors, then like tend to cluster with like, and so homogeneous mixing across, again, a scale of a state might not be the appropriate assumption. Or if you make that assumption, then you just need to make sure that when you interpret the results and present them to especially policymakers, that assumption is explicitly stated. Confidence in our assumptions, that we're making the correct assumptions or that they're reasonable assumptions, will play into the degree of uncertainty of a model. And this is the variability in results, and it can affect how far into the future you're able to forecast. That being said, I think models have quite a bit of value. The value of models is that they are often really the only tools available to us when we are dealing with unknowns or when experimentation is too risky or too time-consuming to conduct. Their models are tools, and these tools enable projections, forecasting. They often guide policy by quantifying phenomena, and so justifying control measures such as our stay-at-home orders and our social distancing guidelines. There are several critical benefits to the use of modeling. The first, of course, is informing policy. Many of our current measures have been based on the information that comes from models. A huge limitation of our response at the beginning of this, of course, was testing. Our testing capacity as a state and a nation, and probably on a global scale, was not prepared for a pandemic. And so in the absence of testing and understanding what the true rates of transmission were at the juncture, models were really the only way for us to, quote unquote, see transmission under certain scenarios. And this has also been a way for us to sort of justify implementing stay-at-home and social distancing orders. The second model benefit is informing the public. So Governor John Bel Edwards has used this, I think, in really good ways. He's compared where we are today with our social distancing and stay-at-home orders with where we could have been had we not implemented these measures. I think just having two different lines on a graph from two different model outputs has been extremely valuable, especially as people become more and more antsy at being stuck home and start thinking about going back to work. So there's a popular saying in epidemiology right now is that if in a year when you look back and think, well, nothing happened, well, that's the point. So because we're at this sort of, this isn't so bad stage, though I'd argue that those people should go sit and talk to a healthcare worker, it's absolutely critical to remind people where we could have been had we not taken any action or had we relaxed too soon. And so models will enable us to quantify the differences in those two scenarios. Another critical utility for model is their future role in designing clinical and field studies to assess the efficacy of therapeutics, vaccine candidates, or even natural herd immunity. But these models will be subject to the same limitations and caveats previously outlined. And a lot of these limitations can be ameliorated with appropriate data from randomized studies aimed at assessing true exposure rates and infection rates in the population, as well as the longitudinal dynamics of potentially protective antibodies. So, but like molecular and medical tests, which cannot in and of themselves create a treatment plan, that's why we still very much need clinicians. Models will benefit from multi-sectoral input to tailor them to capture the important phenomenon from each sector. So we're talking about government, society, public health, and others, because as we learned, COVID is not just a public health issue, it's a very complex social issue. And so models can worked with each of these sectors to identify the data needs from those sectors so that with this multi-sectoral effort, we can create a positive feedback loop that will become a very powerful tool in our response. So in summary, our models are very useful. They have some limitations, but these limitations can be overcome with more and more data. Models are our first line of defense when dealing with unknowns like COVID-19. And at this juncture, when we're looking to return to life after COVID, models will be a critical part of that planning process. But the most critical part of that will be just where's the data going to come from? And so that data is going to need to come from the availability of tests for not only positivity rates, but also antibody and immunity. And so I think that's what Dr. Baines is going to talk to us about right now.
Hello, everyone. Thank you, Dr. Christofferson. So I'm going to talk briefly about testing to measure both virus, and it's not really the whole virus, it's really the virus's RNA and antibodies. So this is a typical scenario from a typical patient based on data that we have currently available for COVID-19 infection. So at day 0, you see at the bottom are the days. We have no antigen, which is the blue line. So as you progress through a typical infection, the antigen levels, and this will vary from individual to individual, so this is just hypothetical based on averages. About day 14, we will have the highest level of antigen. The more antigen or RNA that's detected in these tests, the more infectious a person is. So on day 0, of course, they're not infectious. On day 1, they're a little bit infectious. Day 5, infectious as well. And we don't know actually the threshold when they become infectious to infect someone within 6 feet or 10 feet or anything like that. This is just antigen levels. So the patient responds in a very interesting way, which I don't have time to go into, by making antibody to this infectious insult. And the patient first starts making an antibody called IG. IG stands for immunoglobulin, and M. And that is a transient antibody. It only lasts from about 7 to 21 days. But it's very useful to know about IgM because that indicates, by definition, a recent infection, right? Because it goes away by 21 days. So if you have an IgM positive, that's a person that was infected recently, and that's very useful in the epidemiologic data and would be put into models that might help us understand both the transmission of the disease and also how it's going through a population. Later on, these IgM molecules actually switch in their type from IgM to IgG, and IgG is the red line. And that is our long-term antibody. That lasts for many, many years, potentially. And our antibody tests measure antigen, they measure IgM, the tests measure antigen, IgM, and IgG, but these are different tests. So this is a diagram for the antigen test, and you've seen this on TV. People get swabs. Sometimes it's an oral swab, sometimes it's a nasopharyngeal swab. And what happens in the laboratory is the swab contains virus, and the virus is essentially all the parts that are not interesting to the test are eliminated by extraction. So we end up with an RNA, which is the viral RNA. And using an enzyme called reverse transcriptase, we turn that RNA into double-stranded DNA. Then we put that as the first step. And some of these tests are done in a single step, some are done in 2 steps. So this would be the first step in a 2-step test where we make our DNA. And then we have very specific primers and probes. And the design of these is very important because we don't want to have these primers or probes bind to other viruses that might be in that swab. We want them very specific for SARS coronavirus 2. So those are designed, and frankly, they've been published. The best design has been published and is used by many, many labs. So this is very standardized and is easier to control than some of the antibody tests I'll talk about later. So that goes into a machine essentially with a exogenously added DNA polymerase. And that DNA polymerase not only extends the primer and makes a copy of the DNA, but it also degrades the probe. And the probe is cleverly designed. One end of the probe, and these are very short, just a few nucleotides long, one end of the probe is a quencher, and one is a fluorescent molecule. So when you separate those two, the fluorescent molecule is then freed in the tube, separate from the quencher. So you get a fluorescent event, which is then measured and read out on a fluorimeter or a spectrometer, and then we get a positive result or a negative result if there was no separation of the probe from the quench. So when you think about tests, and this is being bandied about in the popular press, each test, whether it be the PCR test we just went through or the antibody test we'll go through in a second, has a measurable sensitivity and specificity. And the consequences for getting back to work of these measures can be profound. We want both of these to be about 100%, but there are very few tests actually that have 100% sensitivity and specificity. So think about this. So sensitivity is the number of true positives. That means the people who are actually secreting virus divided by the number of positive tests that the test gives. So there are a subset of tests that are false and they read falsely negative. So this is the error in sensitivity. Error is the false negative rate. So a false negative people on the RNA test, if they're allowed back into the workplace, they constitute a risk for everybody else in the workplace. We thought they were negative, but they're actually secreting virus. So they might actually infect other people, even though we thought they were negative. So sensitivity, we really want that correct. We want that accurate. We want that 100%. Specificity is the number of true negatives divided by the number of negative results from the test. So that is false positive rate. So basically the error is a false positive rate. So if you have a test that is not very specific, you're basically going to have a lot of people who think they have the virus, but actually don't. So a false positive on this RNA test, if you use that test to determine whether someone is going to go back to work, you're basically erroring. If you have a lot of false positives, people would be incorrectly excluded from work. They would not be shedding virus, but we think they are. So these numbers are really important about if we're thinking about how we're going to use these tests in order to go to work. And I would argue what we really need is 100% sensitive, 100% specific tests that can be done very quickly before people go to work, preferably at home. That doesn't exist, but that would be the perfect world. So antibody tests, remember, we're measuring IgM and IgG, and that's very useful. A lot of the antibody tests will measure both independently. So this is a typical, these come to your physician. This is a rapid antibody test, and these tend to be a little less accurate than if you were to get a blood draw and send it to a lab. This concept is used in pregnancy tests, for example, and other tests that can be done right in the clinic without having a blood draw. So little pinch of blood goes on that sample pad, and these Y-shaped things are antibodies. The brown ones are IgG, and the pink ones are IgM. And within the cassette is already loaded a antigen. We have COVID-19 as the antigen here, and it's attached or conjugated to a colloidal gold, which actually will elicit a color if it's concentrated enough and has an appropriate substrate. So the test comes with sticky antibodies that will actually collect the patient's IgM and IgG on different lines in the test. These are preloaded, and then there's a control line. So all you have to do, the beauty of these tests is you basically prick your finger, the antibody goes into on the sample pad, and by capillary action, buffer transports the plasma to the right, and it engages all of these different things. So what will happen is that if the patient is positive, we will have a certain line at the M line, which was where the IgM antibody was made, or where it was stuck, and we have the G line, which will show IgG. Now, the issue of this is that we have a lot of these tests on the market, and they're coming on very quickly. There are only 4 that are actually approved by the FDA, so they've gone through substantial investigation by the company. The Abbott test is a test strictly for IgG, and that requires a blood draw, so it has that disadvantage. But the beauty of it is it's 100% sensitive and about 99.6% specific. So it meets one of those criteria. It's very accurate that I talked about, but it can't really be done easily in the clinic, or you have to wait several days for the results. CellX has a rapid test. That's FDA approved. It's about 93.8% sensitive and 95.6% specific. And that measures both IgM and IgG. ChemBio has a serological point of care test, which means it's a rapid test. They don't tell us actually what at least I couldn't find it, what the specificity and sensitivity are. And Ortho has an antibody test that runs through their specialized system. It can do a lot of tests quickly, about 150 tests in an hour. It's sensitive, but not very, about 83% sensitive and 100% specific. So the thing about antibody tests are they're very important unknowns. And these are really critical. This data will become part of the lexicon very soon. But right now, there are critical questions that we don't know. So if you're going to use antibody tests to screen people to see whether they can go back to work, ideally what you would want is for the virus or the disease to cooperate so that we would have everybody who's antibody positive is immune and can't get infected again. We don't know that. We don't really know if these IgG positive people are immune at all. Presumably they are immune. If you look at SARS and MERS, people who recovered were more resistant to infection and certainly more resistant to disease. Another issue is how long does this immunity last? So with SARS and MERS, which are related viruses, but this virus, this particular one, COVID-19, has thrown a lot of, it's been a lot of surprises, and I'm not surprised. I wouldn't be surprised if there were more that the virus will throw at us. But if you just use those as a reasonable example, that immunity lasts between 1 and 3 years. Now, if you have a population that's constantly getting exposed in small amounts, that immunity may last longer because every time you get re-exposed if you have IgG, you get what's called a secondary or amnestic response. And the response actually with antibody is much faster, and the antibody tends to be more effective. So repeated exposures actually improve your likelihood that you will be protected. But again, all of this is an unknown. And the final question is, do IgG positive people secrete virus? I think it's very dangerous to assume that people who have decent IgG levels, and I should say that this rapid test is not quantitative. So we're not actually measuring levels, we're just measuring whether it's detectable or not. Are they secreting virus? So that will be very important to know. Right now, we've been very surprised by we call this serology because IgM and IgG comes from serum usually. Usually you let the blood clot and then you spin out the clot, and what's left is called serum. So we call this study of antibodies in serology. So if these IgG positive people secrete virus, that would be very bad. I will say, however, that what's becoming obvious is that many people are seropositive that apparently had no clinical signs. So there's two interesting studies, one in Los Angeles County. About 4% of people are positive, yet only about 1% actually showed any clinical signs, and fewer than those were in hospital. So we instantly went, if you start detecting people who showed no clinical signs that were actually exposed to the virus, your mortality rate dramatically drops, because up till these antibody studies, we were only basically testing sick people, which have a higher likelihood to succumb from the disease. So what's the mortality rate in Los Angeles County is now dropping to about 0.3%. New York, about 15% of the samples. Now these samples are concentrated in New York City, but they're all also in upstate New York. About 15% of samples are antibody positive. And that is a dramatic difference between those who are actually antigen positive. And now the mortality rate in New York, which had a very high one before, about 4 or 5%, is 0.6%. So the good news is that the virus is probably actually exposing more people than we thought, and fewer people are getting disease. The bad news is it's still a pandemic, and it's still causing significant morbidity and mortality in the population. So that's what I had. The next speaker is Dr. Diaz, and I leave it to him.
Share screen. My topic is going to be investigational treatment options for COVID-19. And the outline of our discussion will be the use of repurposed drugs, and these were drugs that were designed for other uses. We'll talk about some investigational new drugs, some adjunctive drugs and techniques, and then some experimental drugs that are in investigation. We'll talk about immunoglobulin therapy, and you have a good background in that, thanks to Dr. Baines' discussion of immunoglobulin G. And then we'll talk about where we are with vaccines, and then I'll bring up some frequently asked questions if we have time to go through some of those questions. But on the right side of the screen, there are some qualifications that we need to accept before we get started. And that is, number one, there are no proven effective drugs or other therapies for COVID-19 right now. There is the fact that an understanding of the molecular biology of SARS-CoV-2 was based on an understanding of the SARS-CoV-1 virus, which caused epidemics about 18 years ago. So at least we know the viral life cycle. So that's important, and that's the topic of our next slide. Although most of the drugs that I'll talk about are approved for other diseases, none of these new drugs are currently approved by the FDA for the treatment of COVID-19. They can be used by physicians, or you can request the compassionate use of an investigational new drug. And we'll talk about that too. Finally, some of the recommended new therapies are now being tested in randomized controlled trials, which is very important to see if they work or not. Some look very promising. Others don't look as promising as we thought originally. And then finally, effective new drugs for COVID-19 can be evaluated for their efficacy and safety and prescribed for treatment much faster than vaccines can. And we'll talk about vaccines at the end of the presentation. Now, this is an important slide that I'd like to take you through individually. And this slide is actually a diagram of what happens in the lungs. And you can see the SARS-CoV-2 virus, if you can see my cursor, and it is covered with these spike or S proteins. And it is searching for a receptor to attach it or to anchor it to the epithelial cells lining the alveolus of the lung. So that's the host cell for the virus. While it's looking for these receptors, it could be potentially attacked by chemicals that are secreted by specialized white blood cells called macrophages. And we know that some of these macrophages can be important in attacking the virus before it can enter the host epithelial cell of the alveolus. So this is an early area of prevention. If we can prevent the attachment of the S protein to its ACE2, angiotensin II receptor. There are also some other proteins like this one called TMPRSS2, which is a transmembrane serum protease that's also required for entry. So we actually have drugs that can attack the ACE2 receptor. We actually have drugs that can attack this TMPRSS2 protein. And some of these drugs are monoclonal antibody drugs, and some of these drugs, like this one called chemostat mesylate, that's not available in the United States, but we have a similar drug called nafomostat mesylate, and they can prevent binding. So that's the first type of drug. We can keep that virus out of the alveolus. Another new drug that you're going to hear even more about is this one here called Arbidol. Arbidol has another name. It's called umifenivir, and it's a close relative of oseltamivir, and anything that has vir at the end is an antiviral drug. But you probably recognize oseltamivir as Tamiflu, which is the influenza drug. Well, it turns out that Arbidol is effective against influenza and against SARS-CoV-2. So this is going to become a very important drug as we get into the flu season, and we'll be facing cases of both COVID-2 or COVID-19, as well as cases of influenza. Now what happens when the virus gets into the host cell, it begins to uncode itself because it's preparing to make new copies of itself in a process called replication. And we can attack the virus again intracellularly using some of these drugs called lopinavir, ritonavir, darunavir. Now these drugs are called protease inhibitors, and they're used to treat HIV or AIDS infections. And they work intracellularly, and we can use these drugs too, and they look very promising. There are also some other drugs, and you can see them here in the middle, called chloroquine and hydroxychloroquine. These drugs were developed to treat malaria, although most malarias now are resistant to these drugs. And these drugs can prevent both fusion of the SARS virus, and they have some other intracellular mechanisms, so they can reduce the intracellular response to the virus and might be beneficial. These drugs are now undergoing trials, but when we use these drugs to treat malaria, we only gave the drugs once a week. To treat COVID-19, we have to give them every day intravenously, so that we're now beginning to see some adverse effects from some of the antimalarials, particularly cardiotoxicity. Now, finally, before the virus can actually replicate and produce new virus that leave to infect other alveolar epithelial cells. There are some other drugs like remdesivir, which is looking very promising. It's not available now. It's still an investigational new drug, but we have some existing drugs called ribavirin that might be effective as RNA polymerase inhibitors that prevent the last step of the virus from replicating itself. So you can see, because we know the life cycle of the SARS-CoV-2 virus, we can develop drugs to attack the virus before it enters the alveolus and once it's within the alveolus. So a combination of these drugs could be very beneficial at treating the disease. This next slide discusses the potential for experimental treatment of SARS-CoV-2 disease or COVID-19 disease with an experimental drug called the human recombinant soluble ACE2. In this case, we're actually producing in an animal model a drug that can actually bind to the virus before it attaches to the membrane of the epithelial lining cells of the alveolus in the lung. So this is looking very good. It's been tested in small trials in humans with acute respiratory distress syndrome and pulmonary hypertension, but these have been very small trials. It works in a mouse model, and it works in vitro models of human blood vessels and kidney cultures. And we're now recognizing that patients with COVID-19 can have significant cardiovascular disease. They can have clotting within peripheral arteries. They can also have kidney failure because of the widespread predominance of ACE2 receptors. So if we can make this drug in the laboratory called HRS-H2, we can actually trick the ACE2, we can act as a decoy, and the virion, the SARS-CoV-2 virion, will attach to the recombinant ACE2 and not to the membrane-bound ACE2 to get into the lungs. So this is very exciting research that is ongoing right now. Now, the next question is, what about a vaccine? Well, we said earlier when we were discussing qualifications, it takes a long time to develop vaccines. And we'll talk about the timeline for vaccine development in a minute. There are about 115 vaccine candidates right now. 78 are currently in development, 63 are exploratory, and you can see in the bar graph on the right the ones that are being explored, the ones that are being confirmed, the ones that are in preclinical investigations in animal models, and then you can see at the very far right of the bar graph, only 4 are in clinical trials in human beings. So let's talk about those. And these are the 4. If you look at the column on the far left, you'll see all the different types of vaccines. And at the top, you see the live attenuated vaccine. We're not making too many live virus vaccines anymore because we want to be able to give vaccines to patients who are pregnant and patients who are immunocompromised. So it takes a long time to develop these live vaccines. So we're not doing too many live virus vaccines. This is like the mumps, measles, rubella, chickenpox vaccine. But we are developing some of these viral vector non-replicating vaccines, and they take a while. They take about a year to develop. And we're developing some of these protein or peptide-based vaccines, and they also take about a year to develop. All of these 4 are in clinical trials. What's exciting are the bottom 2 vaccines, and these are nucleic acid vaccines. One is aimed at the RNA, and the other at DNA that has to be again, we would have to transcribe that, but both of these vaccines can be produced very quickly within 6 months or a little bit longer. Some may be available to us as early as September. It looks like the Moderna vaccine is much further ahead than all the other vaccines. I'm not sure where the Inovio vaccine is, but it's very exciting that we've got some vaccine candidates that are already being tested in humans, certainly in Seattle and in other areas of the country. So we expect to have vaccines, hopefully, relatively soon. At least some of these fast-producing vaccines. So my last slide, I know we're reaching the end of our discussion, are a number of questions for you to ponder. And if we have time, we can look at these and go through some of the answers. And I will ask our moderator, Dr. Crespo, whether she wants to entertain some of these questions based on the time that we have remaining.
You can go through the questions and give us your thoughts on them.
Okay, thank you. The first question is going to be, have any medical therapies been confirmed to date? The answer is no. The next question is, should hydroxychloroquine, which is one of the antimalarials, and azithromycin, which is the familiar Z-Pak, zithromycin antibiotic used for strep throat, can they be used in combination, and have they been used in severe cases? And the answer is yes, but you may not want to use them in combination because they both have cardiotoxic potential, and in combination they can cause cardiac arrhythmias. And this has happened in some patients in the intensive care unit. Next, if I'm taking a high blood pressure pill, like an angiotensin converting enzyme inhibitor, or like lisinopril, for example, or an angiotensin receptor blocker like losartan, should I stop taking that drug because we know that these drugs can upregulate the ACE2 receptor, which is the one the CoV-2 virus likes to attach to? The answer is absolutely not. It's much more difficult to take care of your blood pressure if you stop taking a medication that you're doing well on. Next, what about immunomodulators like steroids and monoclonal antibodies? At this point, unless there's an indication for steroid therapy such as septic shock or acute respiratory distress, we usually recommend staying away from steroids because they further immunosuppress the patient. However, the monoclonal antibodies, like the ones we talked about and we showed in the viral life cycle, might be very useful, but we would have to use them early along. The same holds true for hyperimmune plasma, which Dr. Baines discussed, because what we want, if you're a survivor and your blood test or your serum test shows that you are IgG positive, we want your IgG because we could use your IgG to attack the SARS-CoV-2 virus before it enters the alveolus. But if we're using hyperimmune plasma containing IgG from people who survived COVID-19, we've got to use it early in the disease. That is, before the virus can get into the alveolar cells. Which of these repurposed drugs appear to be most efficacious? Well, the ones that have been used so far have been the antimalarials like hydroxychloroquine and chloroquine. But maybe some of these anti-HIV drugs could also be very useful, and they're being studied right now as well, called lopinavir, ritonavir, which is a combination HIV drug used for heart therapy. Which drugs are available now? Well, I have a whole list of drugs that are available right now: hydroxychloroquine, chloroquine, azithromycin, lopinavir, ritonavir. We have a number of drugs that we can use. I haven't discussed all of the potential drugs that we could use, some of which are commonly prescribed, such as rosuvastatin or Crestor, and even ivermectin, which is often prescribed by veterinarians for scabies and skin infections on animals, and also lice infections in humans. So this looks like a very promising drug as well. Who should enroll in clinical trials of new therapy? Certainly anybody who's in the ICU, who's either on a ventilator or at risk of going on a ventilator for risk of respiratory failure. Everything we can do to keep a patient off a ventilator would be very important, because the case fatality rate once you're on a ventilator is extremely high. You're no longer in that 1% range. You're now in a double-digit range. Finally, what are the major risk factors for all of these repurposed drugs, drugs that were designed for other uses? The problem is COVID-19 disease can affect the liver. It can cause abnormalities in liver function tests. It can also affect the heart and predispose patients to cardiac arrhythmias, serious cardiac arrhythmias. if you add drugs that can already affect the liver, if you add drugs that can already affect the heart and be cardiotoxic, you get the additive effect of cardiotoxicity from the drug and from the disease. So this is the greatest risk factor that some of these repurposed drugs that we're now using for COVID-19, because we don't have a specific COVID-19 antiviral drug yet. These are the major risk factors. So these are some of the frequently asked questions that I thought you might be interested in discussing. Thank you.
Thank you so much to all our speakers. We hope that you have found this webinar interesting and informative. We will do an update as circumstances merit. In the meantime, I'm gonna share with you some additional resources that you may use. Please visit these websites for additional information. And if you have specific questions for today's speakers, you can visit our webpage at the Department of Environmental Sciences, and we'll make sure to direct your questions to our experts. Thank you for viewing this webinar, and make sure to share it with others who may be interested.